Vaughn Cooper. Go ahead, Vaughn. Vaughn will do the introductions.
Yeah, thanks, Charlie. Thanks for the opportunity to introduce my two colleagues from Pitt's Center for Evolutionary Biology and Medicine, or CBAM, Daria Van Tyne and Matt Solba. They're in the Division of Infectious Diseases here at Pitt within the School of Medicine and within the Department of Medicine and the School of Medicine. Delighted to call them both close colleagues here. They're both at the vanguard of studying the evolution of pathogens in the clinic in near real time. And what makes them, I think, so innovative is first, they start with experience of gaining deep understanding of evolutionary processes during their doctoral and postdoctoral training. Daria working in enterococci at Harvard, and Matt studying an array of pathogens, particularly Staph aureus at Penn. Matt is a physician scientist. Daria is a basic scientist, but they are both really collaborative. And what sets them apart today here at Pitt and broadly in our field is their ability to apply this background of understanding of, what the important questions of pathogen evolution are, with their experience and sort of insight of selecting exactly the right samples to collect, how to collect them, and what's the best technology, both in terms of sort of genomics and microbiological methods, but also bioinformatics and analytical frameworks to study those samples. So a lot of our problems, for instance, in studying pathogen evolution is that we study them sort of as researchers, sort of opportunistically in retrospective fashion. And these two investigators sort of are thinking ahead. And so they're really studying how pathogens evolve in real time using the best methods. Just another comment, in hearing about the work that they're going to present today and in reading the review that they've provided in conjunction with this talk from PLOS Pathogens, I think it's really inspired, at least from my perspective, by foundational work by Bruce Levin and Jim Bohl, which inspired me when I was a graduate student. And what Bruce and Jim pointed out is that pathogen evolution during infections is often really short-sighted. And so although they are adapting, it is often ultimately to no end, not to an end that's going to benefit the species as a whole. So that's one arm of their talk today. Matt and Daria's work builds upon this significantly and adds a lot of meat to this hypothesis, as well as considerably more nuance. So with that, I'll turn it over to, I think, Matt first, followed by Daria. Looking forward to it. Thanks.
All right, thanks for that introduction, Vaughn. That's perfect for what we're going to be talking about today. Give me a second while I get my screen up and shared here. Can you see the slide?
Looks great.
Okay, perfect. All right, so yeah, so the title of our talk today is Bacterial Evolution During Human Infection, Adapt and Live or Adapt and Die. And in the first part of the talk, I'm going to really talk about the comparison of in vitro experimental bacterial evolution and contrasting that with the challenges of applying that framework to studying evolution during clinical infections. And then I'm going to hand it off to Daria, who will talk about a lot of practical considerations in analyzing this. And then both of us are going to, and as well as a framework that we've developed to kind of think about this. And then both of us will use examples of data from our own lab to illustrate a lot of the take-home points. And so I'll get going. So, in order to compare and contrast in vitro experimental bacterial evolution that takes place in the laboratory and try to contrast that with clinical infections, I thought a nice introductory point would be to do a brief overview of one of the more famous in vitro experimental evolution systems. And so Many on the call will be familiar with this, but if you're not, there's this experiment called the long-term evolution experiment that was set up by Richard Lenski in the late 1980s. So briefly, what Lenski did is he set up 12 replicate E. coli cultures more than 30 years ago. And each of these replicate E. coli cultures have been passaged daily into fresh media for literally the last over 30 years ago. This is still ongoing. Every day, 1% of the culture is put into fresh glucose-limiting media, and that culture expands about 100-fold, or about 6 to 7 generations per passage cycle. And this has been going on now for more than 75,000 generations. In basically this same environment where the bacteria just eat the glucose and then move on to the next day. The power of this really comes down to the fact that you can sample the population at any point that you want, and you can cryopreserve that and put it in the freezer and access those samples at any time that you want. So you can resuscitate the culture, study the living bacteria and their phenotypes, and then with the advent of whole genome sequencing, you can now analyze the genetic makeup of these time points with great precision. And that has really offered an amazing ability to study evolutionary dynamics over time in this incredibly simple system. And so there's a lot to unpack from an experiment like this, but I just want to talk about 3 major takeaways from this in lessons learned that we, in ways to analyze this kind of data that we then apply to the study of clinical infections. And it really sort of garners the framework for how I think Daria and I think about studying evolution in a very complex system like a human being. And so the first point that I want to make that these experiments and others like it have shown is that evolution has examples where it's repeatable. And so because there's independent replicate cultures, one observation that is often made is that the same traits and genetic alleles that make up those phenotypes will independently arise within replicate cultures. And so that's a phenomenon called parallelism. And what's neat about that is it tells you that there's a lot of evidence that this is not just a random stochastic thing, that there's probably an actual biological adaptation occurring. And that can be quantified in terms of how many replicates it does and things like this. And so that's a point that I'll harken back to as I describe clinical data. The second point is that evolution is dynamic, which is sort of self-evident in that phrase. But the real point is that what this experiment shows is really the rate in how many times the rise and fall of new genotypes happen. And one of the surprising things is that even after 75,000 generations, this is still happening. And so in the next slide, I'll show you a nice picture of what I mean by that and a couple other take-home points. The third thing that I want to talk about is how the environment can drastically change the temporal dynamics of when a new trait is going to emerge. And so one of the classic examples of this from this experiment is that after about 30,000 generations, if you can see on the bottom there, I've kind of labeled this, a new trait emerged from these E. coli. They gained the ability to eat citrate from the media. Now, this took many years in this experiment for this trait to arise. But what I want to point out is that others have shown that if you take bacteria and expose them to different conditions where you chronically nutritionally deprive them, you can actually have this trait emerge within less than 100 generations, just emphasizing that the environment and the conditions at which selection is taking place really can influence when specific traits can emerge and how fast they can emerge. And we see that kind of stuff happening in clinical infections as well. Okay. So next is so we can actually picture and have a picture in our head of what dynamics look like in these types of experiments. This is a figure from one of Richard Lenski's publications where this is called a Mueller plot. And this is the first 20,000 generations of one of these replicate cultures. And so what you're looking at here is each of these color blocks represents a genotype that was present in the culture. And the height of it on the y-axis of that color block is proportional to its relative abundance within that culture. And as you move from left to right, you're going through time in this experiment. And what you can see is that there are successive times when new genotypes arise and take over and replace previous genotypes. And this is happening all the time. And so that's one of the take-home lessons here. And if you focus your attention sort of to the middle, around 10,000 generations, you can actually see there's two dominant clades here. There's one in green on the top. There's like an orange-red clade on the bottom. So the first thing I'll point out is that these things have really taken over the culture in general, which is evidence that there's probably a lot of adaptation going on to the media. And so there's probably biologically interesting, real adaptive traits that have arisen here. But the other thing that I'll point out is that when this red-orange clade takes over the culture, it completely extinguishes the green one. And so even though there's adaptive alleles in here, they can go away and they can never come back again. And so this is just to make the point that some adaptive mutations are going to live on, but they're, for a variety of reasons, some adaptive mutations are going to die and they won't be seen again. And this is happens in even a simple system like this. As Daria will talk about, in the context of a clinical infection, the reasons that an adaptive mutation may go away can be a lot more complicated and are worth thinking about. Okay, so now tacking to the study of clinical infections. So we can think of clinical bacterial infections as naturally occurring evolution experiments if we have the right conditions. So if you imagine a patient coming in sick with a bad bacterial infection, you can imagine that that bacterial population may be relatively large at first, but as their immune system kicks in and maybe we're giving the patient antibiotics, we could expect that that bacterial population will decrease over time because of that selective pressure. And that there might be genetic diversity available within that setting to select for mutants that get enriched in the population that either are selected for stochastic reasons or may be selected because they have a beneficial advantage. But nevertheless, there are mutants that could be enriched in the population. And if we have a situation where we can sample that bacterial population from the patient over time serially, then we have the analogous situation that I just described in the in vitro setting. And so below here, I'm showing you just one example from my lab of a patient that we studied like this. This patient had what's called persistent MRSA bacteremia, meaning that they had a really severe Staph aureus infection that dipped into their bloodstream, and we could culture the organism out of their blood in this patient for 3 weeks straight. We had the opportunity to collect 15 different isolates over this time period. And so each column in this plot to the right here is a different clone that we cultured from this patient. And then we applied whole genome sequencing to look for variation. Each row in this plot is a genetic variant. So you can think of the blue here as sort of the ancestral state, and then the red blocks containing the mutation listed as a genetic variant. And so I'm showing you here just to point out the analogy to the in vitro situation, but also to point out that when we do an experiment like this in a patient, we get mutations. And then the first thing that we have to ask ourselves are any of these mutations biologically relevant, right? It could be that they arose in these populations and detected for reasons that are just random. And so that oftentimes you find these things and you're now scratching your head. And so in the next slide, I want to harken back to this idea that evolution is repeatable. So in the in vitro setting, evidence for parallelism was obtained because the same trait happened in replicate cultures. In the clinical setting, we can view different patient infections as different replicates. And so they're imperfect replicates, but at least they have the same type of bacterial infection, the same infectious syndrome. And so in this case, I was studying a persistent MRSA bacteremia. And so this is data that we published earlier this year, but we analyzed 20 patients with this disease and similarly collected a whole series of mutations. And we looked for evidence of parallelism. One of the ways to do this is to take we took all the non-coding or the non-synonymous mutations that we found in this data set across these 20 patients. And we just asked a simple question: are any of these mutations happening in any particular gene more often than you would expect by chance? And so if you imagine spreading 63 mutations across 2,600 genes in the genome, most genes are only going to have 0 to 1 mutations. And so if you start finding a gene that has 3, 4, 5, that's very statistically improbable and suggests that something else is going on rather than just a random stochastic process. And so this is called a gene enrichment analysis. And so on the x-axis here, you have every single gene in the genome, and on the y-axis is a probability that the number of mutations in that gene occurred by chance alone. And you can see that we uncovered this CYTZ gene, that we actually uncovered 8 independent mutations across these 20 patients, which was a very good evidence for parallelism, suggesting that these mutations were biologically relevant. Then after we gain evidence like this, it's still not the nail in the coffin for Daria and I. We like to attack and make sure that we can actually prove that these things have a phenotype. And so what we found is that all the mutations that we uncovered here, this gene encodes the citrate synthase gene, completely destroyed the ability of this enzyme to do its job. And so these were all mutations that reduced citrate synthase activity significantly, if not to zero. And then what we could do is we could take those clinical isolates and we could revert the CYTZ allele back to the wild type state. And we studied antibiotic phenotypes. And what we uncovered is that these mutant CYTZ alleles conferred an antibiotic evasion phenotype known as antibiotic tolerance. And so that's just an example of how you can take a bunch of clinical infections, a lot of data, and then zero in and find out a biologically relevant phenomenon that can explain why we're observing this gene happening, mutations in this gene happening so many times. All right. The second point I want to make is that evolution is dynamic. And again, this is sort of self-evident in that phrase, but the real question is do we see dynamics play out during these short timeframes? For persistent MRSA bacteremia, these infections only last 1 to 2 weeks. That we're studying. So it's a very short time scale, and it's, curious of whether or not there are dynamics at play here. For the most part, myself and others that study clinical isolates during bloodstream infection, we typically have 1 clone per time point, and that makes it very, very difficult to estimate allele and genotype frequencies as a function of time. And so this is actually some unpublished data that I just got last week, and in an effort to do a better job at this, we've now characterizing patients with more genomes. And so this is a patient who is bacteremic for 7 days, but we have 60 genomes that describe this infection, with 10 independent genomes at every single time point. And so this allows an opportunity to estimate genotype frequencies with much better precision. And so on the plot here, what I'm showing is we uncovered a handful of genotypes here, and each of these colored lines represents a genotype. And you can see that there's this genotype in light blue that starts off at 0 frequency in this patient, but then is enriched over time up to 100% frequency. And then that is supplanted by new genotypes that then end up taking over the population. And as it turns out, these new genotypes actually occurred on the backbone of this previous genotype, and they share a common allele. And so in the Mueller plot to the right, you can see that this clade now basically overcomes the population almost to fixation. And so we're able to actually study dynamics in sort of the analogous fashion of what I showed you in the in vitro scenario. And this, when we see something rise like this, it suggests that there's that there may be an adaptive advantage again. The third point that I want to make is that the environment can drastically change temporal dynamics. So this is a similar analysis now of a different patient where I'm plotting allele frequency versus time. This patient was characterized with 95 separate genomes, about 10 genomes per time point again. And one thing I'll just point out is that we see some different alleles rise in this patient, and then something really drastic happens when the addition of rifampin antibiotic is added to this patient's antibiotic regimen. We see that those alleles crash and go down to 0. And at the end of this infection, when we analyzed it, we see that the entire population is dominated by genotypes that contain 3 different alleles, all with mutations in this RPOB gene. The reason that that's significant is because RPOB is the target of the drug rifampin, and RPOB mutations are very well known to cause rifampin resistance. And so what's likely happening here is that the reason that this rapidly arises and overtakes the population is because these are all these genotypes are all rifampin resistant now. And so when we change the environment with antibiotics, we can see drastic changes in the dynamics. Harkening back to, again, what can be observed in vitro experiments. And so just to summarize here, in vitro principles and methods can be applied to clinical infections, but of course, you really need a special type of clinical infection to do this. And so it has to be an infection where you can do serial sampling over time. You have to be able to detect de novo mutations, and you need an infection and a time frame where you would expect something to actually happen. And so that's a lot of the challenges. So this type of analysis really is only suited to really a minor subset of the types of infections that we see in patients. And so that's one point. And then I'm just going to close by sort of going over some of the main things that we think about when we're talking about the complexity of analyzing infection like this in a human. And so the way to look at this infographic, if you will, is that these little culture vials denote the in vitro situation, and these little people denote the situation in a clinical infection. And so one thing is that the genetic variation in an in vitro system is well defined, whereas in people, we often don't know what the mutation rate is or what the population size is, and those things can be changing through time, and it's not clear to us what that is, and so that's a challenge. The other thing is that in a closed in vitro system, horizontal gene transfer is either absent or you know exactly what vectors are available for transfer, whereas in an in vivo system, this is a completely open system, and it can be a source of important genetic variation depending on what you're studying. Perhaps more interesting is the different types of selective pressures. And so in people, there can be tissue microenvironments with host passage and competition for nutrition at all kinds of different anatomical sites. Antibiotic exposures may vary between different anatomic sites from a patient. You have the immune system to contend to, which is a major selective force. Different spatial structures, including biofilms. Patients often have catheters or prosthetic heart valves or prosthetic joints, and biofilms those can be sources of infection with biofilms. And so spatial structure is something that could be very important in these infections. And then you can have polymicrobial infections too, and it's possibly that the makeup of the community structure of all the different microorganisms may also influence evolutionary trajectories. And so they're very complicated systems, and it's challenging and sometimes daunting to think about. But I think this is exactly what is so exciting about doing this type of work, because you can begin to what the goal is here is we find examples of evolution that and then we try to do experiments in the lab directed at trying to figure out exactly what those selective pressures are to really illuminate what the underlying biology is and uncover new things. And so this is why we're so excited about this type of work. And so that's everything that I'm going to talk about. I'm going to hand it over to Daria now.
So I put in the chat that if anyone has any questions specific to your talk, Matt, now is a good time for a few of them, and then we'll turn it over to Daria. So, I see Joe. Hey, Joe, would you like to raise ask one of your questions in the chat?
Sure. Yeah, Matthew. First, great talk. Really interesting work. You know, people can tell how interesting I think it is by all the inane things that I typed in the chat during the talk. But one of the things I think you guys could consider doing it is, in my mind, the degree of parallelism you're going to see in your systems is going to be a function of how similar the patients are. Now, there are going to be a variety of variables that could play a role there, ancestry particularly being one of them, but also biological sex and age. And that would be, great example for a large NIH study to see how much parallel responses depend upon patient characteristics. I would think less so for the use of the antibiotics themselves, but for the immune response, I think that those factors would play a big role. So anyway, great work and keep it up, guys.
Yeah, Joseph, I can't agree anymore. I think as more and more people are getting into the space and more publications are coming out, and we're seeing the variety of types of things that are evolving in patients, I think it's becoming more and more clear that there are syndrome-specific signals even within particular infections. And it's not very well defined yet. And certainly the patient is I agree with you, is probably probably the dominant driving force that affects the trajectory of what's happening in these patients. And so, yeah, we're very excited to try to tackle those questions.
Michelle Blythe, I see you have your hand up. Want to go ahead?
Yeah, I'm an infectious disease physician. And I was just curious what your thoughts were as far as if you would expect some of these, if you would expect this to be different in infections that are actually contagious. So, for a lot of ID, it's not really contagious stuff that we're dealing with. It's, as you study, Enterococcus and Staph aureus and stuff of that variety. So would you expect kind of different patterns, I guess, in organisms that are actually, like COVID or something that is actually contagious?
Yeah, absolutely. So Daria will be, I think, discussing sort of how we think about the transition of within a single patient evolution and what the implications are for infections that are actually transmitted. I'll just briefly say, like, in an overall system, there's for Staph aureus particularly, there's really great work. One person that comes to mind is Bo Shopson, who has studied community outbreaks of MRSA skin infections, and has had many samples that have broken out across an entire community. And in that setting, you can see evolution occurring during transmission events. And so there's definitely sort of a spatial geography transmission component of this. And then there's also the study of evolution within single patients as well. And both can be true. So if it is an organism that can be transmitted from person to person or is contagious, then both scenarios absolutely apply.
Thank you so much.
Thanks for the question. And I think that's a really good transition for Daria. So Daria, take it away, please.
Thanks so much. Yeah, let's I'm going to take up where Matt left off. So I'll just start by thanking the organizers for the invitation to spend this hour with you. And Matt sort of laid a great framework. I'm going to take it forward a little bit to talk a little bit more about how he and I think that we can or should be studying this process of bacterial evolution in vivo during human infection. And so, as Matt alluded to, the biggest challenge we face in trying to do these kinds of studies is sampling. So what can you actually collect? What should you actually collect to do these kinds of studies? And so obviously, just to state our goal, if it's not already obvious, we want to try and identify de novo adaptive mutations and then characterize the adaptive traits that those mutations might confer. And that's to try and, study this evolutionary process in the kind of messy ecosystem of the human body, but also to gain new insights about the biology of these organisms, and with our sort of clinical hats on to try and devise improved therapeutic strategies to treat patients suffering from these infections. And so as Matt said before, I'll reiterate again, so sampling in this scenario is very limiting, but it's still possible. So we are thinking of different patients with the same infection as even with all of their variability between patients, they can nonetheless be biological replicates and allow us to sample the same process happening over and over again. If the patients are chronically infected, especially if they're admitted to the hospital, we have access to them and we can sample them repeatedly over time. The types of samples that we collect are almost always specimens that are collected as part of routine clinical care of a patient. So Matt and I both do work studying bacteremia, so bloodstream infection. So to diagnose a patient with bacteremia, you have to collect a sample of their blood and then test that sample to see if there are bacteria in it. And so that process, generates a sample that the clinical laboratory once they're done processing the sample and determining whether it has bacteria or not, they're going to throw that sample away. And if we can be sitting there, at the trash can with our hand out, we can collect that sample when the lab is done with it. And so medical centers like ours have robust research and infrastructure that'll enable us to collect these remnant clinical specimens to sort of fuel our research projects. And I will just note on another practical IRB side, sequencing bacterial genomes and doing these studies, even though we're asking questions about evolution within the patient, because we're only studying the bacteria, we're only querying the bacterial genome, we actually don't need to get the patient's consent to do so. All the samples are de-identified. We collect them and process them so that they're free of human genetic material. And so when we ask questions simply about the bacteria evolving over time, we can collect a lot of patients and we don't actually have to enroll them in our studies to do so. And I want to acknowledge, and as Matt and I were writing our review article, we looked at the literature and we saw that a lot of the prior work in this area has largely focused on high frequency and, quote, longer lived mutations. So because for practical reasons, you can actually sample chronically infected patients and get multiple samples from them over time. Most of the prior literature in this area has focused on chronic infections and how bacteria evolve over relatively long time scales. And a lot of the prior work, as I'll show on the next slide, has been in, for example, CF patients with pulmonary infections caused by different types of bacteria. And the opportunity that Matt and I saw here is that, we are aware of lots of patients that have acute infections that are not chronically infected. They have acute infections with things like VRE or things like MRSA, and we wonder about adaptation in that setting. And we simply are asking with our research, can we observe evidence of adaptation even on short timescales? And that sort of led to the kind of framework that we proposed in our review article. So we are interested in perhaps lower frequency, but definitely much shorter lived mutations and their potential that they could uncover new biology and lead to new discoveries. And so with sequencing costs continuing to drop, Matt was talking about sequencing, tens of isolates from each patient, or sequencing the entire population as a pool. As the feasibility of collecting more and more whole genome sequencing data, increases, we think it's increasingly possible to study these rare mutations in a practical way. And so that's all to sort of build up to the two kind of scenarios that Matt and I have conceived of to sort of, we used it as a sort of a useful structure to frame this review article that we wrote. But we basically were thinking about adaptations that bacteria could make while they are infecting people and how we might think about those types of mutations. And so we made these two buckets and we called one bucket adapt and live and another bucket adapt and die. And so the first big difference between these buckets is simply the time scale. So adapt and live mutations, we think of them as being long-lived on the months to years time scale. We often find them associated with chronic infection or perhaps asymptomatic colonization. They may or may not be transmitted to other people, but the opportunity for transmission is there just because they're longer lived. They may be at higher frequency. They persist for a longer time. And some of these mutations may in fact enhance pathogen transmission and enable their propagation over a longer time scale. And then in contrast, so adapt and die mutations would be just the opposite of all of that. So these mutations are not long-lived. They may stick around for days or weeks, but we're not going to see them over the months to years time scale. They would arise due to their selective advantage, in that moment or, in that short time frame for the bacteria, but then they might be rapidly extinguished. So in the case of an acute infection, this could be because the infection resolves, the immune system comes in and clears out the infection. We give the patient antibiotics and that cures their infection. It could also happen that these adaptations occur at body sites that that don't enable transmission. They don't enable the pathogen to get into another person. And the idea of evolutionary dead ends is one that is sort of a common current through my work, the work in my lab at least. And I'll show an example in just a couple of slides. So we think these adapt and die mutations are much less likely to be transmitted to other hosts. And because of that, they would have to arise independently in each newly infected host. And so this fact that, or this idea that that evolution would have these adaptations would have to occur over and over and over again, the idea of evolution on repeat, also is a way that we could identify these adaptations and study them further. Okay, so we came up with a couple of cartoons to sort of visualize these two buckets, the adapt and live and adapt and die. And so this is the sort of visual aid we developed for the adapt and live scenario. So we could imagine a patient that has a pulmonary infection. So these are the little circles here are the different bacteria causing their infection. So that patient's initially infected, maybe they get sick, so they get some treatment with antibiotics, or maybe they have hopefully at least partially functioning immune system. So the immune system was going to come in and try and clear the infection. And so antibiotics, host immune system, these are going to apply selective pressures on the population and cause adaptive changes over time. And so if we came back again and sampled that patient again, we might find that the pathogen that's causing their infection is related to the ancestral pathogen from the initial infection, but it might also be have acquired mutations. And if those mutations are adaptive, they may have facilitated things like antibiotic resistance, or a broad kind of umbrella process that we call pathoadaptation, which is the sort of long-term evolution of pathogens during chronic infection. And if we go forward in time some more, then some of those bacteria from that patient may transmit and go on to infect other patients, or they may establish a chronic infection in that patient. And so if we came back to that same patient again and did a third sampling and compared the bacteria in that infection with what we sampled prior, we might find again that the same bug is there, but that it's acquired additional mutations and has undergone additional adaptation. And so the examples for this process that we can find in the literature are largely, as I've said, pulmonary pathogens, Pseudomonas, Burkholderia, and largely in patient populations that are prone to chronic infection and that also have frequent healthcare system interaction so that they can be sampled. And so cystic fibrosis patients and the evolution of these pathogens in these patients is a big part of the existing literature on this topic. And there's also a fair bit of work also in tuberculosis about sort of the evolution of resistance, transmission of resistant TB. And the common, so if you look at all these studies and you ask sort of what happens, what's the commonality between them, some of the common adaptations that we see are evolution of antibiotic resistance, these pathoadaptive mutations that affect how the bacteria and host basically talk to one another, the host microbe interactions, immune evasion, decreased virulence, things like that. And then the last thing that you frequently observe is the appearance and emergence of hypermutators. And to harken back to the long-term evolution experiment, some of those replicate lineages also became hypermutator lineages. And it seems that during the process of chronic infection, one way that the pathogen may try to adapt is to become a hypermutator. So this is the adapt and live scenario, but perhaps more, I don't know if this is more provocative or not, but the new thing here, I suppose, would be this adapt and die scenario. And so, so for the purposes of the review and for the discussion, we thought of two kind of dominant examples of infections that might fall under this adapt and die scenario. And the first one is the emergence of vancomycin-resistant Staph aureus. So this is a process by which in a polymicrobial infection, you'll have vancomycin-resistant enterococci, which are my favorite bug, and methicillin-resistant Staph aureus, which are Matt's favorite bug. And they'll find each other and they can undergo horizontal gene transfer. And so when this happens, it always happens that the VRE, the vancomycin-resistant enterococci, give their vancomycin resistance plasmid to the staph. And that makes the staph turn into Versa, or vancomycin-resistant Staph aureus. And so these are very scary because they would be very, very difficult to treat and also very virulent. And the concern is that these Versa might become epidemic. They might go on to be transmitted to other patients. But it turns out that becoming vancomycin resistant is extremely costly for Staph aureus. So for Staph to make its cell wall in a way to be that it would be vancomycin resistant, in the absence of vancomycin pressure, it is like a really, really bad way to make the cell wall. So these Versa, in the absence of antibiotic selection, are so unfit that we have literally not ever seen evidence of transmission of Versa from one patient to the other. It means that when Versa evolves, it always happens within a patient, and the infection is always confined to that single patient. We don't have follow-on transmission because the evolved variant is so unfit, it cannot be transmitted. And then the second example is an Enterococcus specific example. So Enterococcus, as well as other GI tract bacteria or pathogens that live in the GI tract, Enterococcus is very well evolved to live in our GI tract. The bacteria have spent hundreds of millions of years evolving to be GI tract commensals. And we now find ourselves with lots of patients that get Enterococcus infections, but those infections happen outside of the GI tract. So Enterococcus infections happen when the bacteria migrate from the GI tract to extraintestinal sites. And one of those sites is the bloodstream. And so if we imagine the two different places where a patient that has a VRE infection might have bacteria, they would have VRE in their GI tract, and then they'd also have VRE in their bloodstream. And the VRE here are these little Diplococcus guys who are a little bit pointy. And the point of this figure is to show that if this patient has VRE in their GI tract, they can transmit that VRE to another patient. But if that patient gets VRE in their bloodstream and the VRE adapt, those adaptive changes that happen in the bloodstream of this patient are actually not going to be transmitted to the next patient because there's no bloodstream transmission of VRE. All the Enterococcus transmission that happens via the fecal oral route. So if there's either a fitness barrier or an anatomic barrier to passing these adaptations on to another patient, that's going to be a scenario where we're only going to see them, within a single patient, and they would fall into this adapt and die kind of bucket. And as I said before, we think of these infections as kind of evolutionary dead ends. And so I was going to take a couple of slides to just show a real life example of this from a paper that I developed as a postdoc with Mike Gilmore, looking at multidrug-resistant Enterococcus faecalis. And this paper feels like ancient history because we published it before the pandemic, but it came out in 2019, which is really not that long ago in Science Translational Medicine. But this paper centered on a collection of E. faecalis that were sampled from patients with bacteremia all the way back in the mid-1980s. So right before Richard Lenski was getting ready to start the long-term evolution experiment, at a hospital in Wisconsin, they experienced a widespread outbreak of multidrug-resistant E. faecalis bacteremia. So a colleague at the time who was working in this hospital actually collected all of these blood isolates and stuck them in the freezer. And then 30 years later, when I was a postdoc, we sequenced their genomes and studied them. So there were samples available from 53 patients. And when we sequenced their genomes and compared the genomes to one another, we found that all 53 of these patients were infected with bacteria that were very closely related to one another. And so if we looked at their accumulation of mutations over time, we could find that there was a steady accumulation of variants, of a relatively small number of variants, I should say, over about a 3-year time period. And from that information, we concluded that these patients were all infected with this closely related, quote unquote, clonal outbreak lineage. Of course, it's not clonal because it has, individual isolates will have mutations that distinguish them from one another, but those mutations are relatively small number of mutations. So we think that this was a classic sort of fecal oral transmission, patient to patient of E. faecalis. But we sampled all these patients from their blood. And so if we think about, the different lineages that, or the movement of the bacteria here, looking at these variants that were shared between where we found them in multiple isolates in the data set, we could infer what we think the genotype of the transmitted lineage might have been. We did not sample any Ephecalus from the GI tracts of these patients. So all we can do is infer based on what we sequenced of isolates from the blood. So this figure is just meant to show in a schematic way the sampling of all these different isolates over time. And if we imagine that the GI tract population of Enterococcus that was transmitting between these patients may have been collecting these shared variants as it transmitted over time. What we actually sampled were isolates from the blood, and these bloodstream isolates, these lines that are sort of spokes off of the wheels here, are meant to show variants that were unique in these isolates that we sampled in the blood, but that weren't found in other blood isolates. And it's these. So the shared variants tell us that this lineage was an outbreak and was transmitting between patients and acquiring mutations as it was transmitted. But these singleton variants, where we see them only one time in one isolate and never again, are what we might consider, or where we might look for adapt and die variants. And so we looked for either gene level or locus level parallelism in this pool of singleton variants that we had. And we actually identified a couple of different places in the genome where we found evidence of parallelism. So I'm just going to summarize them very briefly because a lot of this work was not focused on identifying them, but actually figuring out the biology that could be driving these mutations. And so two of the signals that we saw were locus level parallelism, where an operon, a collection of genes in the bacterial genome, was repeatedly and independently mutated. And so the first one was the cytochrome operon, where there are 4 genes that together encode a cytochrome. And we saw 6 different mutations in different isolates that all fell in this operon. And so we asked, why would mutations in this operon, what benefit might the bacteria have if they had mutations in this operon? And with some in vitro work using a isogenic deletion mutant, we found that this operon gives the bacteria the ability to respire in the presence of heme. So if the bacteria are grown in media, they will grow to a certain optical density. And if we give wild type bacteria heme in that culture, they will grow to a higher density. They can generate more of themselves if we give them some heme as well. And this ability is absent if we knock out this cytochrome. So the ability to grow more in the presence of heme is because of this cytochrome operon. But I mean, that's fine. So the bacteria can't grow very well in the presence of heme if they have this locus mutated. But there's other studies in the literature suggesting that when bacteria like Enterococcus or staph turn on respiration, they become more susceptible to neutrophil-mediated killing. And so we took our wild type of mutant strains and we competed them in the presence of whole human blood. And we found that the mutant isolate was able to outcompete the wild type isolate in this assay. And so it seemed that these mutations that we observed in the cytochrome operon may have helped the bacteria better resist killing in human blood, for example, by decreasing their susceptibility to neutrophil-mediated killing. And then a second signal that we observed was in this EPA locus. So this is a long operon that contains a ton of different genes that work together to make cell surface associated polysaccharides, so sugars that the bacteria put on their surface. And we didn't explore the impact of these mutations in this study, but there are other studies in the literature showing that when you change these genes in the EPA locus, if you mutate them, you change the composition of the surface polysaccharides, and these changes confer things like decreased both antibiotic as well as bacteriophage susceptibility. And then the last signal was like a very overwhelming signal from this data. We observed that there were 18 different isolates that all had different mutations in a single gene, which was a transcriptional regulator. So this these signals were great, and they seem to make sense biologically. This signal really sort of hit us over the head with the strength of the signal. So why would there be 18 different mutations in this single transcriptional regulator? And to make a long story short, I spent years of my life as a postdoc exploring the molecular mechanism underlying these GNTR regulator mutations. And at the end of the day, it boiled down to these mutations changed the transcriptional program in the bacterial cell to cause changes in the cell surface associated polysaccharides. And doing so gave the bacteria decreased susceptibility to killing with both beta-lactam antibiotics like ampicillin, as well as lysozyme, which is an enzyme that chews up the bacterial cell wall. And these mutations also increased bacterial survival in an infection model. Here we used a mouse abscess, but if we competed a GNTR mutant with an isogenic wild type strain, we found that the mutant could grow better in this infection model over. And so that's all I wanted to say about that. The paper is published, and you're welcome to explore it further if any of that was of interest to you. But to just conclude, so we have some time for discussion at the end. Matt and I study how bacteria evolve in vivo during human infection, and we want to do that to try and learn more about the biology of the organisms and try and identify targets for therapeutic intervention. Obviously, there are lots of practical considerations that limit our ability to do these studies. We do our best to overcome these. Personally, I think it's fun to study these sort of messy, complicated systems. It feels like a big sandbox to me. And that I think the novelty of our framework is the idea that beneficial traits could arise during infection and be short-lived. That these things might be challenging to study, but we think that if we do study them, that we could gain new insights into how bacteria adapt to these environments within the human host. And so with that, I'm going to conclude the presentation by thanking all of you and looking forward to some discussion. I'm going to open the chat window now to see what happened during that.
All right, if you've been holding your questions, now's the time. I know, Charlie, you had one, so if you want to kick it off, or
Yeah, sure. It really goes back to earlier in the talk, mostly, I think. Matt, you brought up, it goes back to Matt's talk, I think, mostly. You brought up antibiotic tolerance. I wonder if you could describe how that's different from antibiotic resistance. How that's kind of operationally defined or mechanistically defined. Say a little more about that.
Yeah, sure. So antibiotic resistance is really defined by a particular type of assay. And importantly, the assay that is used to define resistance is one of that measures growth inhibition by an antibiotic. And the key to this is that it's measuring growth inhibition, and there's no measurement of life and death of the bacteria. It's just growth inhibition. And so generally what's done in, say, a clinical lab is you will take bacteria and expose them to different serial dilutions of antibiotic across usually twofold concentration differences, and you just grow them up and you see what the minimum amount of antibiotic is that inhibits growth. And then that concentration gets deemed the, quote, minimal inhibitory concentration, or the MIC. And that is an extremely useful parameter clinically, because it really predicts, if you're using a treatment antibiotic and the MIC to that drug is elevated, it really predicts treatment failure. And so that's frank resistance, basically meaning the bacteria can grow in the presence of a high concentration of this antibiotic, right? Yeah. Okay, so less explored is this idea of antibiotic tolerance, which I think the majority of the evidence show this probably does impact patient care, but it's very difficult because the assays are difficult, and we don't have any ways to get around it anyway. So, so tolerance is really a life-death thing. And so in the strictest sense, tolerance is a decreased rate of killing. So it applies to bactericidal antibiotics, where if you put the antibiotic on a bacteria and measure a time kill curve, the amount of death that's happening as time goes forward, and you measure the rate of killing, a tolerant strain is going to have a slower rate of killing. Although if you were to measure the minimal inhibitory concentrations in a growth inhibition assay, it would be identical. So it's a life-death thing as opposed to a growth inhibition thing.
Yeah, so I think of like resistance is, I ask the question, can the bug grow in the presence of this antibiotic? Can it grow or not? If it can grow, it's resistant. But for tolerance, I ask, at a high concentration of antibiotic, does the bug die? Or how quickly does it die? Or after I've killed what I can, is there a fraction that remains that would be tolerant? And so, yeah, the framework is different, the assays are different, and we're measuring basically different phenotypes, even though it kind of gets put under the whole umbrella of antibiotic susceptibility.
Great. Thank you. Appreciate that.
Yeah.
Great sort of foundational question. And just to editorialize, we've found a lot of similar adaptations, both in other studies of infections and in animal models of infections, to proceed by mutations that produce tolerance, not so much frank resistance, or at least a mix of both. And that mix of both is maybe a nice handoff to Wynn's question in the chat. Wynn, are you still on? Can you ask your question sort of in person?
Thanks, Vaughn.
Yeah, thanks for, just wanted to say another Pittsburgh connection. Good to see you.
Hey, good to see you.
Yeah.
Yeah, it was because of Dr. Graves's suggestion in the chat that potentially if you had adapt and die mutations, they could like shift where you are in phenotype space and make some adapt and live mutations possible. Are there any systems available to get at that more directly? Like are there some systems where you see both of these patterns and you could, I don't know, look at evolution in a broader scale for these kinds of patterns?
Yeah, that's a great question. I mean, I think anything is possible. And of course, like these buckets that Matt and I made, they're just buckets, right? It's just sort of a framework for thinking about it. And I really like the idea that, adapt and die mutations give maybe a short-term advantage, and that opens up space for an adapt and live mutation to take off. I could imagine that like in the studies that we do, we could find evidence of that if we saw, repeatedly independent mutations in a gene, and then, a haplotype that takes over that carries one of those. That would be evidence of maybe the two types of mutations working in concert.
Yeah, Matt, are you aware of any instances of sort of combinations of traits becoming transmissible, whereas mutation 1 is maybe local, but mutation 2 is maybe more global, and consequently the two then transmit?
I can think of, so in tuberculosis, which is pretty well studied for this, I think a lot of the resistance mutations that have arisen and have been discovered to be transmitted between patients, some of those actually, as an individual mutation, do carry a pretty high fitness cost. And there's a thought that you need a compensatory mutation that relieves that fitness cost, but still allows for the resistance trait. And that, so that you really sort of need this double mutation in order to permit efficient transmission. And so I guess that would be one example, because you could imagine that resistance may, under selective pressure in an individual patient, you might get the resistance mutation first, and then that's the substrate on which you get an additional compensatory effect, which actually enables transmission. So I think there are examples like that in tuberculosis, for example.
I see we are at just past the hour, so I want to be respectful of everyone's time. I'm not seeing any other hands up. So I guess I'll take this time to
Well,
thank.
actually, Vaughn, I did have my hand up really quickly.
Oh, sorry.
It's
Go
all
for it.
good. And I was going to say, maybe the place to look for the Wrightian ship that we're talking about is in the LTEE, because it's still adaptation. It's not infection. But if there's any place where you would find that evidence, it would probably be in the LTEE based on that 2017 paper. So you could look there and establish the principle, then it might be easier to see an infection. That's all I want to say.
Thanks, Joe. Well, I was just going to say, well, I want to just thank our speakers again, and thank Trisem and the team for continuing to keep this great opportunity rolling for evolutionary medicine.
Great. Thank you for hosting, Vaughn, and thank you, Matt and Daria, for participating, Becca for helping to organize. Thanks everyone for tuning in. We really appreciate it. We love to see so many people here and see all the discussion in the chat.